Welcome. My name is Yongjun Gao. On behalf of my co-authors, in this webinar, I will discuss and demonstrate how to use the clinical relevance approach described in the FDA's MAPPS for establishing impurity limits in drug substance specifications in drug master files. What's MAPP? MAPP stands for the FDA's Manual of Policies and Procedures. MAPPs are federal directives and documentation of internal policies and procedures. MAPPs are required by law and are made available to the public to make FDA a more transparent organization. FDA's MAPPs can be found at FDA's website. Today, I will be talking about two impurity-related MAPPs. The first one is MAPP 5017.2, establishing impurity acceptance criteria as part of specifications for NDAs, ANDAs, and BLAs based on clinical relevance. It became effective on May 1, 2020. The second one is MAPP 5310.7, acceptability of standards from alternative compendia. It became effective on October 13, 2017. The purposes of these two MAPPs are twofold. The first purpose is to provide guiding principles and approaches for establishing drug substance and drug product impurity acceptance criteria for non-mutagenic impurities in NDAs, ANDAs, and BLAs based on the consideration of clinical relevance. The second purpose is to clarify what types of data and information are needed when establishing impurity acceptance criteria in drug substances and drug products. Establishing impurity limits in drug substances represents one of the common challenges for both the pharmaceutical industry and the regulators, especially when ICH guidelines don't apply. Here, I will be using 6 real case examples to illustrate how to use the clinical relevance approach from the two FDA MAPPs I just mentioned to establish impurity limits in drug substance specification in drug master files. Case 1. The acceptance criterion of a specified impurity may generally be established at up to the ICH Q3A qualification threshold if there is no other information to support the need for a lower limit. For example, for the drug substance dapagliflozin, currently there is no compendial monograph. Its ICH qualification threshold is 0.15% based on the maximum daily dose of 10 mg. So, the acceptance criterion of each specified impurity in the drug substance can be set at the ICH qualification threshold of 0.15%. Case 2. The acceptance criterion of a specified impurity should be set at lower than the ICH qualification threshold if there is a compendial limit related to safety, which is lower than the ICH qualification threshold for the impurity. For example, for the drug substance atorvastatin, which is used for producing the popular cholesterol-lowering drug Lipitor, the USP specified impurity atorvastatin 3-deoxyheptatenoic acid has a USP limit of 0.10%. Although its ICH qualification threshold is 0.15% based on the MDDO 80 mg, the acceptance criterion of this impurity in the drug substance specification should be set at a maximum of 0.10% instead of the ICH qualification threshold of 0.15%. Case 3. The acceptance criterion of a specified impurity may be established at more than the ICH qualification threshold if the drug substance has a USP monograph, or it doesn't have a USP monograph but has other compendial monographs, and the monograph acceptance criterion is greater than the ICH qualification threshold. For example, for the drug substance isomeprazole sodium, it doesn't have a USP monograph, but it has an EP monograph. In the EP monograph, EP specified impurity D is controlled at 0.2%. Even though its ICH qualification threshold is 0.15% based on the MDDO 240 mg, the higher EP limit of 0.2% can be considered acceptable. It should be mentioned that the impurity should be verified. There is no apparent risk. For example, there is no structural alert. The assessor usually should verify that the monograph limit has been justified by some type of data in an approved product. Case 4. The acceptance criterion for a specified impurity in a DMF referenced by an ANDA or a 505(b)(2) NDA product may be supported by a side-by-side comparative impurity analysis for the proposed product and the RLD. Just to give you an example, the drug substance clindamycin palmitate hydrochloride has a USP monograph. However, USP doesn't control any specified impurities. On the other hand, ICH qualification threshold doesn't apply due to that this drug substance is a semi-synthetic antibiotic. In this case, since neither compendial monographs nor ICH guidelines apply, the DMF holder can perform a side-by-side comparative impurity analysis of 3 batches of the proposed drug substance and the RLD. For example, if impurity 1 was found at a level of 0.5% in both the RLD samples and in one applicant's samples, the acceptance criterion of 0.5% for the specified impurity 1 in a DMF can be generally justified. It should be emphasized that the comparison should be conducted on multiple batches of the proposed product and the RLD. The same analytical method should be used for the analysis, and the analytical method should be validated to demonstrate it is suitable for its intended purpose. Case 5. The acceptance criterion of a specified impurity may not be set at the ICH qualification threshold if there are known safety data for the impurity based on its structural class. For example, the presence of a structural alert for mutagenicity. For instance, for the drug substance difamipridine, it has a USP monograph. In the USP monograph, impurity 3,5-dibromo 4-aminopyridine is controlled at a maximum of 75 ppm. Even though the qualification threshold is 0.15% based on the MDDO 20 mg, the acceptance criterion of this impurity in a DMF should be set at a maximum of 75 ppm instead of the higher ICH qualification threshold of 0.15%. Case 6. The proposed acceptance criterion may be supported by data or literature demonstrating that the impurity is a significant metabolite of the drug substance. For example, for the drug substance macitentan, it has no USB monograph or other compendial monographs. The ICH qualification threshold is 0.15% based on the MDDO 10 mg. According to the literature in clinical pharmacokinetics, impurity ACT132.577 is a significant metabolite of the drug substance macitentan, and it has no structure alert. Therefore, the limit for impurity ACT132.577 can be set at the ICH qualification threshold of 0.15% in a DMF. It should be mentioned that in some cases, the clinical significance of a metabolite may be subject to pharmatox and all clinical pharmacology consults. In summary, the FDA's impurity-related MAPPs provide guiding principles and approaches for establishing drug substance impurity acceptance criteria for non-mutagenic impurities as part of specifications for DMFs based on the consideration of clinical relevance. These policies can serve as useful tools for both the drug applicants and for the FDA assessors, helping accelerate the drug approval processes. Thank you for your interest. You can send a question regarding this poster to the following email by February 15th for inclusion in the poster Q&A session on March 4th. A following-on webinar for both poster and oral presentations will be held on April 9th. Questions can be sent to the About email by March 19 for the webinar. You can also refer to the following titled presentation on March 4th for additional information. Thank you again.